Okay, good morning, guys. My name is Alexander, and my project was about oxygen production in Dolphin Island plants. So let's begin. The purpose of the project was to determine which of the 7 plants that I tested that are native to the Dolphin Island Sea Lab dune systems would produce the most oxygen in a bicarbonate solution. So the recent rise in greenhouse pollution or greenhouse effects linked to human consumption of fossil fuels is a large problem, and plants obviously consume CO2 and release oxygen. So basically, plants are awesome because they do this for us. We release CO2 and they take up CO2 and produce oxygen. So it's like a cycle. And the more oxygen is produced, obviously that's better for us. So yeah, we really need these plants, and that's what this was about. So a little background, photosynthesis is the process that plants do. So they convert CO2 into oxygen and produce ATP. So it occurs in the chloroplast of the plant cells, which is the little like green part in the cell. And oxygen is less dense than water. So if you have oxygen underwater, it'll cause the plants to float. So that was really the basis of my project. So my hypothesis was that the Chinese tallow, scientific name Triadica sebifera leaf, would float to the surface fastest because the density to surface area ratio was very high for that plant. So the plant leaf was really thin and it wasn't as dense, which meant that the way I was measuring the mass, you could get more surface area. So the materials was a 0.2% sodium bicarbonate solution, which is really just baking soda mixed into water. And that provided the carbon. So like I said before, plants use CO2 and they transfer it into oxygen. So the bicarbonate, it provided the carbon that they use in this cycle. And the plant leaf cycle, or the plant leaf samples that I used, consisted of Physalis walteri, which is a plant here called the Walter's ground cherry, and it's native to the area. Rus copalina, which is winged sumac. Triadica sebifera, which I was in my hypothesis, it's Chinese tallow. Ceratiola eryxoides, which is seaside rosemary. Amorpha fruticosa, which is small leaf plant. It's very long and it's got a bunch of small leaves on it. Sereno repens, which is saw palmetto, and Pinus elliot, which is it's a type of pine tree that grows in the maritime forest here. So I used a heat lamp, a syringe, and a hole punch, an analytic balance to mass it, and clear glass jar and a glass beaker. So the procedure consisted of mixing 0.06 grams of baking soda into 300 milliliters of water, and that gave me the 0.2% sodium bicarbonate solution that I needed. I hole punched 5.01 gram samples from each plant, and obviously since some were denser than others, I had to cut it down to get it to that weight. So then I proceeded to inject the plants with sodium bicarbonate, which meant that you basically took the plastic syringe and provided a vacuum with the sodium bicarbonate solution in it, which forced it into the cells. So that would provide the carbon, and then the light, the heat lamp, would provide the solar energy, and that would cause oxygen to form through photosynthesis. So these are some pictures that I took during the experimentation. Up here you see this is Walter's ground cherry. That's one of the leaves that I tested. And up here that's the seaside rosemary. And right here is the winged sumac. So those are some leaf samples. And right here you can see I'm massing it, and it's at 0.01 grams. So this is it before I injected it with the bicarbonate solution. And then up here you can see it's at around 0.023 grams, which is higher because obviously I injected it with the bicarbonate solution. So you can tell that it is in fact saturated with the solution. And in the middle here, this is the vacuum that I formed. So you'd cover the tip right here. You'd cover that with your finger, and then you'd pull back the plunger, which would force the solution into the cells. And right here is how I tested it. There's a heat lamp above this. You can kind of see it right there. And the plants would sit here at the bottom, and the more oxygen they produced, the oxygen bubbles should have adhered to the leaf and caused them to float to the surface. So I basically measured the time it took for them to reach the surface. So these are the results. As you can see, I'm missing some of the results. And this, I don't really have an explanation for it. I'll try my best to discuss that after these results. But right here you can see the Physalis walteri, which is the Walter's ground cherry. It rose to the surface fastest. So this is time in seconds, and these are the trial numbers, and these right here are the leaf samples. So I only got results for 3 of the leaf samples out of the 7 I tested. Okay, so the results show that Walter's ground cherry floated to the surface the fastest, and my hypothesis was shown to be incorrect by the data. However, I believe that the results were incomplete because I think that some of the plants, the leaves were like the leaf surfaces. So you have glamorous versus papillate and coriaceous leaf surfaces. So I think that the oxygen bubbles didn't adhere to them. So for some of them, I actually saw oxygen being produced on the leaf, and they were just floating off the leaf, so they weren't even adhering to the leaf. So I believe that that caused some of my results to be incomplete and inconclusive. I'd like to thank the University of South Alabama and the Emerging Scholars Environmental Health Academy for giving me the scholarship to come here. I'd like to help Tina Miller Way, Dr. Tina Miller Way, for helping me out and getting the project straight. Jenny Cook was my mentor. She helped me out with the project a lot. I'd like to thank Dolphin Island Sea Lab staff for being there and helping out during the process. I'd like to thank Dr. Ishara Ramkasoon for giving me the scholarship as well. Miss Emily Hosford, my science teacher back at Davidson, for introducing me to the program, and Madeline Thomas for sitting around and watching plant leaves like float. So yeah, that's the project. Any questions? Yeah, sure. Um, no, because this is time in seconds. So this is the amount of this is the amount of time it took to float to the surface. So the lowest amount of time would be the fastest oxygen production. So Walter's ground cherry was. Any other questions? No? Okay, well, thank you for listening.